Welcome to The Explainer. Okay, today we are going on an absolutely incredible journey. We're zooming all the way down into the microscopic building blocks of life itself. We're going to uncover the exact chemistry that makes you, well, you.
Okay, let's dive into this and start with the question that might sound kind of strange at first. What actually separates our living, breathing human bodies from the non-living dirt and rocks on the ground? You might think the answer is a completely different set of magical, life-giving chemicals. But the shocking truth? A living cell and the Earth's crust contain the exact same basic elements. Carbon, hydrogen, oxygen, they're in the dirt and they're in you.
So the crucial point here is that the difference isn't what elements are actually present, but their relative abundance. Oxygen is actually the highest in both the Earth's crust and the human body. But elements like carbon, hydrogen, oxygen, nitrogen, and sulfur, what we can call the CHONS elements, they represent a significantly higher percentage of your body weight compared to the Earth's crust. Meanwhile, elements like sodium, calcium, magnesium, and silicon, those are far more abundant in the non-living rock. It's really all about the recipe's proportions.
To really ground us in the numbers, let's look at the cellular mass. If you take a living cell, what's actually inside it? Well, the overwhelming majority, around 80% of a cell's mass, is just water. It is by far the most abundant chemical in any living organism. After water, we have proteins making up about 10 to 15%, followed by nucleic acids, carbohydrates, and lipids in much smaller amounts.
Section 1. Filtering the cellular soup, the making tea experiment.
Now you might be wondering, how do scientists actually figure out what organic compounds are swimming around inside this cellular soup? To understand this, let's use a highly relatable analogy: making a cup of tea. Here's exactly how it works in the lab. Scientists take a piece of living tissue, like, say, a piece of liver or a vegetable, and they grind it up. But they don't grind it in water. They grind it in a weak acid called trichloroacetic acid. This creates a thick paste or a slurry. Then, just like pouring loose-leaf tea through a strainer, they filter this thick slurry right through a cotton or cheesecloth.
And this brilliantly illustrates how we separate a cell's components. We get two totally distinct parts. First, the filtrate. This is the liquid that passes through the cloth and into the cup. It's the acid-soluble cool. These are tiny micromolecules weighing less than 800 daltons, which, is just a microscopic unit of mass, and they mostly represent the cell's cytoplasm. On the flip side, we have the retentate, the stuff that gets caught up in the strainer. This is the acid-insoluble pool, made up of massive macromolecules weighing over 10,000 daltons. These big players represent the larger cell organelles.
Section 2. The Great Lipid Exception, the rule breaker of the cell.
Chemistry always has a plot twist, right? There is one major rule breaker that completely defies this filtering logic we just did. Lipids. Lipids, which make up our cell membranes, are technically very small. An individual lipid molecule weighs less than 800 daltons, so by all rules of logic, it should slip right through the cheesecloth and into the filtrate cup. But it doesn't. When we grind up the tissue, we disrupt the cell membranes. Because lipids have water-fearing or hydrophobic tails, they instantly panic and clump together to hide from the surrounding liquid, forming these tiny clustered bags called vesicles.
Because they clump into these protective vesicles, they artificially act like heavy, massive macromolecules and get caught right in our strainer. This is a massive scientific technicality. It's exactly why biologists say lipids are not strictly macromolecules. They sit in the retentate not because they are heavy, but because they hold on to each other so tightly.
Section 3. The Metabolite Toolbox, Primary versus Secondary.
So, we've filtered our cellular soup. Now let's categorize the organic molecules we've actually found. These carbon-containing compounds involved in the cell's metabolic reactions are called metabolites, and they basically fall into two distinct toolboxes. First up, we have primary metabolites. These are the absolute fundamental building blocks of survival. We know exactly what they do, and without them, the organism literally dies. Think of glucose for energy or amino acids to build proteins. But then you have secondary metabolites. These are highly specialized tools, usually found in plants, fungi, and microbes. The organism doesn't necessarily need them to survive day to day, but they serve massive ecological and, for humans, economic purposes.
Now, what's really interesting about this slide is just how much our modern world relies on these secondary metabolites. Plants produce these chemicals to, say, attract pollinators or defend against pests. But look at what we use them for.
We extract their pigments for colors, we harvest their alkaloids to make powerful painkillers like morphine and codeine, we use their specialized chemicals to create drugs like the anti-cancer compound vinblastine, or even extract polymeric substances to make everyday industrial rubber. It's just wild.
Section 4. Carbohydrates, the sugar pearls. Zooming into monomers and polymers.
Let's shift our focus entirely to one specific, incredibly important primary metabolite, carbohydrates. To truly understand how complex sugars are built in the body, I want you to imagine a single pearl. In chemistry, that single individual building block is called a monomer. But if you take a bunch of those pearls and string them together onto a thread, you create a beautiful pearl necklace. That large connected structure is a polymer. Carbohydrates, scientifically known as saccharides, or just sugars, are classified exactly by how many of these pearls are strung together. A monosaccharide is just a single pearl. It's one simple sugar unit, made of roughly 3 to 7 carbons. If you link a few of them together, say a short string of 2 to 9 pearls, you get an oligosaccharide. And if you string together a massive, heavy necklace of 10 or more sugar pearls, boom, you've built a complex polysaccharide.
Now, if we put one of these individual sugar pearls under a microscope, we see they usually form ring shapes. And the shape really matters. A pyranose ring is a structure that contains exactly 5 carbons sitting directly inside the ring itself. Glucose is a perfect example of this. On the other hand, a furanose ring contains exactly 4 carbons inside its main ring structure, like ribose, which is the sugar found in our RNA. Keep in mind, these sugars might have more carbons in total, but those extra carbons, they just hang on the outside of the ring.
Let's move to and see how this builds in Section 5. Linking the pearls together. The chemistry of the glycosidic bond.
Knowing the shape of the pearl is really only half the battle. How do 2 sugar pearls actually physically attach to one another to start making that necklace? Well, when you place 2 simple sugars like 2 alpha-glucose molecules side by side, they don't just magically stick. To lock them together, chemistry demands a toll. You literally have to remove exactly 1 water molecule, an H2O, from between them. The oxygen that gets left behind forms a super tight bridge between the first carbon of one sugar and the 4th carbon of the other. We call this bridge an alpha-1,4 glycosidic linkage. This removal of water to build a bond is the absolute fundamental mechanism of how your body builds complex carbohydrates.
Think about that for a second. To store energy, to build cell structures, your biology is constantly stringing these pearls together, dropping a molecule of water with every single link. So I leave you with this to ponder. How many millions of these tiny, microscopic glycosidic bonds are being formed, broken, and reformed inside your body to keep you alive right at this very second? It is a literal chemical symphony happening within you every single day. Thanks for exploring with us.